My name is Breanne McAlpin, a fourth-year life science student under the guidance of Dr. Syed Qadri. Today, I'll be discussing my NSERC project where I explore the mechanisms behind reduced red blood cell viability in hemolytic disease. To understand this topic, it is essential to first understand some key concepts. First, let's start with hemin, which is a chemically modified form of heme and a byproduct of its metabolism in the body. Heme is an iron-containing molecule in hemoglobin, which is a protein in red blood cells responsible for oxygen transport. When heme is metabolized, it becomes hemin, also known as ferric chloride heme. Hemin plays a role in regulating the quantity of the blood, but its function can be categorized into physiological and pathological significances. Normally, heme is involved in oxygen transport and hemoglobin regulation. Pathologically, however, excess hemin can lead to oxidative stress, causing cellular damage and cell death. This oxidative damage is particularly relevant in the context of hemolytic diseases, where red blood cells are ruptured through a process known as hemolysis. This destruction of cell leads to an increased release of hemoglobin into the blood, which is then converted to heme. This conversion affects heme's ability to bind to oxygen. In my investigation, I aim to understand how these mechanisms affected red blood cells viability. Human blood was provided from the human bank, was washed and exposed to different temperatures. I tested different concentration of hemin at 1, 10, and 100 micromolars. Following incubation, the cells were washed and stained using two different fluorescent dyes, BODIPI and DCFDA. BODIPI quantified lipid peroxidation, while DCFDA detects reactive oxidant species, where higher fluorescent intensity indicates increased oxidative stress. After staining, the cells were washed and plated, and fluorescence was measured using a plate reader. This allowed for insights on how varying hemin concentration and temperature variation impact oxidative stress and red blood cell viability. In conclusion, my project highlighted the integrated relationship between heme metabolism, oxidative stress, and red blood cells destruction in hemolytic diseases exposed at different temperatures. By understanding how excess hemin contributes to oxidative damage, we can better comprehend the underlying mechanisms of these diseases and potentially develop more targeted treatments. This research not only enhances our knowledge of cellular processes, but also paves the way for future investigation aimed at understanding the effects of hemolytic diseases. Thank you.